Florence, 1348. The writer Giovanni Boccaccio is watching his city die. In the opening pages of the Decameron, he describes what he sees with the precision of a man who cannot look away. The streets littered with the sick and the dead, neighbors abandoning neighbors, parents abandoning children, doctors fleeing the city they were sworn to protect. The disease arrived without warning and killed without logic. A person could go to bed healthy and be dead by morning. The swellings appeared first in the groin or armpit, hard, dark lumps the size of an egg or an apple that the Italians called gavoccioli. Then the black patches spread across the skin. Then the fever. Then, in most cases, death. Within 4 years, somewhere between 25 and 50 million people across Europe, the Middle East, and Asia were dead. Entire villages were erased. Cities lost half their populations. The clergy who stayed to administer last rites died alongside their congregations. The doctors who remained tried everything they knew, and almost everything they tried failed. But here is the question this documentary asks, and it is not the question you have probably heard before. The question is not, why did medieval medicine fail so completely? The question is, in the middle of that failure, what was quietly working? Because some people survived, and some of the things that helped them survive were not prayers, not astrology, not the application of a live chicken to a plague bubo. Some of them were plants, some of them were practices, and some of them, discovered only centuries later, were written into the survivor's DNA itself. By the end of this video, you will understand not just what medieval medicine got wrong, you will understand what it got right, without knowing why. And you will understand how the largest single mortality event in recorded European history left a genetic fingerprint that modern immunologists are still reading today. To understand what medieval medicine was doing, and why some of it worked, you first have to understand what medieval medicine thought it was dealing with. The dominant medical framework in 14th century Europe was not superstition exactly. It was a coherent theoretical system inherited from the Greek physician Galen, who had synthesized ancient medical knowledge in the 2nd century AD, and transmitted to Europe largely through the Arabic translations of the Persian polymath Ibn Sina, known in the West as Avicenna, whose canon of medicine, written around 1025 AD, remained the standard European medical textbook for over 500 years. The theoretical foundation was the doctrine of the 4 humors: blood, phlegm, yellow bile, and black bile. Health was understood as the balance of these 4 substances in the body. Disease was their imbalance. Treatment aimed to restore equilibrium through diet, through purging, through bloodletting, through the application of substances that were classified as hot, cold, wet, or dry, and matched to the patient's particular imbalance. This framework was wrong. Not slightly wrong, fundamentally wrong. It had no concept of infection, no concept of bacteria, no concept of contagion in the modern sense. When the Black Death arrived, the Paris Faculty of Medicine, the most prestigious medical institution in Europe, convened to determine its cause and issued a formal report in 1348 attributing the plague to the conjunction of Saturn, Jupiter, and Mars in the sign of Aquarius on the 20th of March 1345. They were not fringe thinkers. They were the best medical minds in Europe, applying the most sophisticated analytical framework available to them, and they were catastrophically wrong. But here is the thing about a wrong theoretical framework. It can still generate correct practical observations. A physician who believes that garlic is effective because it corrects a humoral imbalance may be entirely wrong about the mechanism, and entirely right about the result. The history of the Black Death is full of exactly this kind of accidental accuracy. People who survived, practices that helped, remedies that had real chemical activity, and not one practitioner in the 14th century who could have told you why. The most striking early example of this is not a plant or a remedy at all. It is a bureaucratic decision made by the city of Ragusa, modern Dubrovnik, on the Croatian coast in 1377. Facing an outbreak moving toward their harbor, the city council passed an ordinance requiring that all incoming ships and travelers wait for 30 days in an isolated location before entering the city. The period was later extended to 40 days, 40 giorni in Italian, the origin of the word quarantine. The reasoning behind this decision was entirely wrong. The city fathers of Ragusa believed in miasma theory, the idea that disease was carried by bad air emanating from rotting matter. Keeping travelers outside the city walls was supposed to give the bad air time to dissipate, but the practical effect was something modern epidemiology would recognize immediately: a cordon sanitaire that interrupted the transmission chain of a flea-borne bacterial infection. Wrong mechanism, correct intervention. Ragusa's mortality during the Black Death was dramatically lower than comparable cities that did not implement quarantine. The lesson was not that medieval people were clever about infection control. The lesson is more subtle and more interesting. That empirical observation, noticing that cities with more traveler contact died faster, could generate effective interventions even in the complete absence of correct theoretical understanding. It is a lesson that runs through every aspect of what medieval medicine got right. Now we come to the plants, and this is where the story becomes genuinely interesting, because when you run the herbal remedies of the medieval plague period through modern phytochemical analysis, the results are not what the history books suggest. The history books suggest that medieval plant medicine was a mixture of folklore and desperation. The phytochemical data suggests something more complicated, that several of the most widely used plague remedies contained compounds with real, documentable antimicrobial activity against the specific bacterium that was killing people. Let's start with garlic. Allium sativum has been used medicinally across virtually every culture in recorded history. In plague-era medicine, it was applied topically to buboes, consumed in large quantities, and rubbed on the body as a preventative. Medieval physicians justified its use through humoral theory. Garlic was classified as hot and dry, and the plague was understood as a cold and wet imbalance. The mechanism was wrong. The result was more interesting. In 1944, the chemists Chester Cavallito and John Bailey isolated allicin, the organosulfur compound produced when garlic is crushed or chopped, and demonstrated its powerful antimicrobial properties. Subsequent research has shown allicin to be effective against a range of gram-positive and gram-negative bacteria. Studies on Yersinia pestis specifically have shown garlic extract to have measurable inhibitory activity against the plague bacterium in laboratory conditions. This does not mean that eating garlic cured the plague. It means that a substance with genuine antimicrobial activity was being applied to infected tissue by people who had no idea what a bacterium was. Next, Four Thieves vinegar. This is one of the most famous plague remedies in history, and also one of the most misrepresented. The legend holds that four thieves in plague-stricken Marseille, or Toulouse, depending on the version, were able to rob the homes of the dying without contracting the disease, protected by a vinegar infusion they had prepared. The formula varies across historical sources, but consistently includes wine vinegar or cider vinegar, garlic, rosemary, wormwood, sage, clove, and various other aromatics. Modern analysis of this formula reveals a remarkable concentration of antimicrobial and insect-repellent compounds. Acetic acid, the active component of vinegar, is a broad-spectrum antimicrobial that has been used as a disinfectant since ancient Greek times and remains in clinical use today. The aromatic herbs, rosemary, sage, thyme, wormwood, contain volatile compounds including cineol, camphor, thujone, and various terpenes that have demonstrated antimicrobial, antifungal, and insecticidal properties in modern research. The insecticidal component is particularly significant. Remember that Yersinia pestis was transmitted by fleas, specifically the oriental rat flea, Xenopsilla chiopis, a preparation that repelled insects applied to the body before handling the sick and the dead would have reduced flea bites, not because anyone in the 14th century understood flea-borne bacterial transmission, but because the herbs happened to contain the right compounds. There is one more remedy worth examining in detail: theriac. Theriac, also called treacle in its English form, was the most prestigious and expensive plague remedy in medieval Europe. Its origins were ancient. It was first developed as a universal antidote to poisons and venoms in the 1st century BC by Mithridates VI of Pontus, elaborated by the Greek physician Andromachus into a formula of up to 84 ingredients, and standardized for European use in the 12th century Antidotarium Nicolai. By the time of the Black Death, theriac had become almost mythological in its reputation. A cure-all whose preparation was a public ceremonial event in major cities, supervised by the civic authorities to guarantee authenticity. Its formula varied but consistently included opium, viper flesh, long pepper, cinnamon, myrrh, saffron, and dozens of other botanical and animal substances. Did theriac cure the plague? No. Nothing cured the plague in the 14th century. But opium, present in substantial quantities in theriac, would have provided genuine symptomatic relief for the fever, pain, and respiratory distress associated with late-stage plague infection. This is not trivial. A patient in less pain, sleeping more, consuming more fluids, has a marginally better chance of surviving a borderline infection than a patient in agony. Theriac did not treat the disease. It may have treated the patient well enough to matter. Now, for what didn't work. And this section is important because the point of this documentary is not to rehabilitate medieval medicine wholesale. The point is to distinguish, with modern scientific clarity, between the things that had genuine activity and the things that were dangerous, useless, or both. Bloodletting. This was the most universally applied treatment in medieval medicine for virtually every condition, including plague. The theoretical justification came directly from humoral theory. If disease was in excess of one humor, removing blood would restore balance. In practice, bloodletting in a plague patient achieved the opposite of what was needed. Yersinia pestis causes a massive systemic inflammatory response and septicemia, a condition in which the body is already fighting desperately to maintain circulatory volume and immune function. Removing blood reduced circulatory volume further, weakened immune response, and accelerated death. Bloodletting killed plague patients faster. The flagellant movement. Between 1347 and 1351, groups of penitents, sometimes numbering in the hundreds, moved from town to town across Europe, publicly whipping themselves in acts of collective penance. They believed that the plague was divine punishment for human sin, and that self-inflicted suffering would atone for that sin and end the epidemic. The movement generated extraordinary popular energy and genuine social disruption. It also spread plague efficiently, as large crowds of people moving between infected communities guaranteed maximal exposure at a time when isolation was the only effective intervention available: the chicken treatment. One of the most frequently cited examples of medieval medical absurdity is the practice of plucking a live chicken and applying its bare flesh to plague buboes. The theoretical reasoning that chickens were believed to breathe through their posterior openings and would therefore draw the infection out is obviously not anatomically defensible. More practically, the bacteria from a live chicken applied to an open, infected bubo would have introduced additional pathogens to an already compromised wound. The treatment didn't help. In some cases, it may have actively worsened the infection. Urotherapy, the drinking and bathing in urine. Some medieval physicians prescribed clean urine as a medicinal wash or drink, based on the belief that properly filtered urine carried healing properties. This belief was not entirely without ancient precedent. Certain components of urine, including urea and antimicrobial peptides, do have documented biological activity. But the net effect of encouraging sick people to drink urine rather than clean water during a period of high metabolic stress and dehydration was negative. What these failed treatments share is revealing. They were not random. Each had a theoretical justification: humoral, theological, folkloric, or pseudo-biological. Each represented the application of available knowledge to an unprecedented problem. The failure was not stupidity. The failure was a theoretical framework that had no mechanism to correct itself when evidence contradicted theory, because the concept of evidence-based correction did not yet exist. The contrast with the effective remedies is stark. The things that worked, quarantine, garlic, insect repellent aromatics, worked because they intervened at the level of actual biological mechanisms, even when their practitioners had no idea that biological mechanisms existed. In 1996, a researcher named William Paxton published a paper in Nature that would become one of the most cited findings in the history of AIDS research. Paxton had been studying a group of people who defied statistical logic. HIV-negative individuals who had been repeatedly exposed to the virus through high-risk contact and had still not seroconverted. They were not using protection consistently. They were not on prophylactic treatment. They were simply not getting infected. Something was protecting them. What Paxton and his collaborators found was a mutation in a gene called CCR5, specifically a 32 base pair deletion that produces a truncated, non-functional version of the CCR5 protein. In its normal form, CCR5 is a receptor on the surface of white blood cells. HIV uses it as an entry point. Without functional CCR5 on the cell surface, HIV cannot enter the cell. People who carry 2 copies of the CCR5 delta 32 mutation, homozygotes, are nearly completely resistant to HIV infection, regardless of exposure. This discovery was remarkable enough on its own. What followed was equally remarkable. Researchers began asking the obvious question: if this mutation confers such powerful resistance to a pathogen, where did it come from? Beneficial mutations don't just appear. They spread through populations because they confer survival advantages. Something must have selected for CCR5 delta 32 in European populations. The mutation appears at a frequency of roughly 10% in people of Northern European descent, far higher than in populations from Africa, Asia, or the Middle East, and it appears to have spread rapidly through European populations within the last few centuries, which is exactly when the Black Death was killing between 1/3 and 1/2 of the European population. The hypothesis, first proposed by researchers including Stephen O'Brien and now supported by evidence published in PNAS in 2024, is that Yersinia pestis may use CCR5 as one of its cellular entry mechanisms in a way that parallels HIV. Individuals who carried even 1 copy of the CCR5 delta 32 mutation may have been partially protected from the most severe plague outcomes. Over the course of multiple plague outbreaks across the 14th, 15th, and 16th centuries, those individuals survived in disproportionate numbers and passed the mutation to their descendants. The mutation was selected for by the plague, spread through the surviving population, and centuries later, in a completely different pandemic caused by a completely different pathogen, turned out to confer resistance to HIV as well. The research village of Eyam in Derbyshire, England, provides one of the most striking pieces of supporting evidence. In 1665, the village was struck by plague and made a remarkable collective decision: to quarantine itself voluntarily to prevent the infection spreading to surrounding communities. The villagers knew they were condemning many of themselves to death. They did it anyway. When modern researchers analyzed the DNA of Aam's surviving descendants, they found CCR5 delta 32 allele frequencies significantly higher than in neighboring villages, consistent with the hypothesis that the mutation had protected a subset of the village's population during the outbreak. This finding does not prove that the Black Death caused the CCR5 delta 32 selection. The academic literature is appropriately cautious. Alternative explanations, including other plague outbreaks, smallpox, and genetic drift, have not been ruled out. But the correlation is striking, the biological mechanism is plausible, and the practical consequence is real. A genetic variant shaped by medieval catastrophe has become one of the most important targets in modern HIV treatment research. Several drugs now in development or clinical use, including the CCR5 antagonist Maraviroc, approved by the FDA in 2007, work by binding to the CCR5 receptor and blocking HIV entry, mimicking the protection that CCR5 delta 32 homozygotes carry naturally. The Black Death did not just kill 50 million people. It may have left a genetic legacy that is helping us fight a 21st century pandemic, and will continue to do so for as long as HIV exists as a therapeutic challenge. Before we close, the caveats. Because this story, like every story in this channel, requires intellectual honesty about what the evidence actually supports. First, no medieval herbal remedy cured the plague. Garlic, four thieves vinegar, theriac, and aromatic herbs had real chemical activity. That activity may have reduced secondary infections, repelled plague-carrying fleas, or provided symptomatic relief. None of it treated Yersinia pestis infection at the systemic level. Without antibiotics, streptomycin, doxycycline, ciprofloxacin, bubonic plague remains a life-threatening disease today. The mortality rate without treatment is between 30 and 60%. With treatment, below 5%. The gap between what medieval medicine could offer and what was needed is enormous. Second, the CCR5 delta 32 and Black Death hypothesis is supported by evidence but not proven. The academic literature is cautious and appropriately so. Multiple alternative explanations for the elevated CCR5 delta 32 frequency in Northern Europeans, including other historic epidemics, smallpox, and genetic drift, have not been ruled out. The 2024 PNAS paper by O'Brien and colleagues strengthens the case but does not close it. Third, in vitro antimicrobial activity is not clinical efficacy. Garlic extract inhibiting Yersinia pestis in a petri dish is not the same as eating garlic protecting a person from bubonic plague. The concentrations needed for in vitro activity are typically far higher than dietary consumption produces in human tissue. Fourth, do not interpret this video as an endorsement of herbal medicine as an alternative to antibiotics for bacterial infections. It is not. The lesson of the Black Death is not that traditional remedies are equivalent to modern medicine. The lesson is more specific, that empirical observation can generate useful interventions even without correct theoretical understanding, and that modern science can identify which historical observations were pointing at real biological activity and which were not. That distinction between what the data shows and what we want it to show is what separates history from mythology. Florence, 1348. Streets full of the dead. Doctors who knew the theory but not the cause. Patients who tried everything and survived on some combination of luck, genetics, and the accidental chemistry of a handful of plants. The people who rubbed garlic on their bodies, burned wormwood in their rooms, and kept strangers at a distance were not practicing medicine in any modern sense, but they were doing something that medicine, in any era, ultimately depends on, paying attention to what worked, even without understanding why. 700 years later, We are still learning what they observed, and sometimes we are finding that they were more right than they knew.